I am Ankita Tiwari, PhD scholar from Department of Pharmaceutical Sciences, Dr. H.S. Gaur University, Sagar, with an interesting and learning episode of B.Pharm on the important unit of antibiotics in Medicinal Chemistry 3, 5th semester, which is scripted by Kapil Jain, working as Research Associate in Reckitt Benikser India Limited, and Pradeep Nirbhavne, PhD scholar in Punjab University. First of all, we'll go through some basic introduction and important terms related to antibiotics, and then in detail, mechanism of action for the drugs. In this chapter, we describe different types of antibiotics. Module 1, Introduction to Antibiotics. Module 2, Classification of Antibiotics. Module 3, Mechanism of Action. Module 4, Beta-lactam antibiotics. Module 5, Aminoglycosides. Module 6, Tetracyclines. Module 7, Miscellaneous antibiotics. And Module 8, Summary. Module 1, Introduction to Antibiotics. Drugs used for treating infectious diseases are called antibiotics, anti-infectious agents, and antimicrobial. All of these terms are basically interchangeable. Antibiotics are essentially natural compounds produced by microorganisms that are capable of inhibiting growth of pathogenic microbes, bacteria. Semi-synthetic antibiotics generally are products that are partially chemically altered versions of antibiotics that are isolated from natural sources. It should be noted that there are compounds produced by microorganisms with antifungal and antitumor action, which also are classified as antibiotics. First of all, we'll understand some important terms related to antimicrobial antibiotics. Selective toxicity. The general concept of antimicrobial action is called selective toxicity, which entails the growth of the infected organism is inhibited or destroyed by certain drugs without harming host cells. High selective toxicity of antibiotics to microorganisms is explained by the unique qualities of the organization of microbial cells, which are principally different from mammalian cells. The nature and degree of this selectivity determines whether the given antimicrobial drug is generally non-toxic in its relationship with mammalian cells, or if it just exhibits certain toxicity on certain mammalian tissues. Minimal inhibitory concentration, that is MIC. A series of two-fold dilutions of a drug in a solution containing a standardized amount of microorganisms are observed. The results are expressed by an index of minimal inhibitory concentration, that is MIC, which is the minimal concentration of drug that inhibits visible growth after overnight incubation. Minimal bactericidal concentration, that is MBC. MBC is determined by the absence of bacterial growth on agar plates that have been re-incubated for more night. This is the lowest concentration of drug that destroys a minimum of 99.9% of the bacterial contents of the test. Bacteriostatic. Bacteriostatic drugs inhibit bacterial growth but do not destroy these organisms in clinically attainable concentrations. It should be expected that the MBC of such drugs will be significantly higher than the MIC. For example, tetracyclines, sulfonamides, etc. Bactericidal. Bactericidal drugs cause death of microbial cells and their lysis at clinically attainable concentrations. For such drugs, the MBC is close or equal to the MIC. Antibiotic resistance. The ability of bacteria and other microorganisms to resist the effects of an antibiotic to which they were once sensitive. Antibiotic resistance is a major concern of overuse of antibiotics. There are two types of resistance, internal resistance and acquired resistance. Internal resistance. This type of microbial resistance is the genetic ability of a microorganism that is coded in the chromosomes and spread to all lines of the given type of microorganisms. Acquired resistance. Acquired resistance means that the given line of a type of bacteria acquired the ability to oppose the given antimicrobial drug. Acquired resistance implies a change in the DNA of the bacteria that results in the appearance of new characteristic features. Such resistance is achieved in two ways: mutation of chromosomes in bacteria or acquisition of new pieces of DNA, that is plasmid, that code for a function of resistance. The biochemical mechanisms of internal and acquired resistances are identical and can be explained as the result of one of the following four reasons. First, inactivation or modification of drugs by bacterial enzymes. Second, formation of an impermeable membrane so that the drug cannot reach the desired region of action. Third, changing the target itself so that the drug cannot bind or have an effect on it. And fourth, development of altered metabolic pathways which permits the effect of the drug to be bypassed. Additive effect, synergism, and antagonism. If the organism undergoes simultaneous action of two antimicrobial drugs, the activity of drugs in combination is equal to the sum of their independent activity is called additive effect. Antagonism occurs when the overall effect of two antimicrobial drugs are less than the sum effect, and synergism occurs when overall effect of drugs are more than the sum effect. Synergism can occur as a result of various mechanisms of action, such as subsequent blockage of the general metabolic pathway or increasing the permeability of bacterial cells. Now let us proceed to module number 2. Module 2, classification of antibiotics. There are several ways to classify antibiotics. In particular, antibiotics are classified according to either principal biological origin, for example, antibiotics developed by certain microorganisms, mechanism of their biological action, for example, antibiotics that inhibit synthesis of nucleic acids, and the spectrum of biological use, for example, antibacterial antibiotics with a narrow spectrum of use, active mainly with respect to gram-positive organisms, antibacterial antibiotics with a broad spectrum of use, antituberculosis antibiotics, antifungal antibiotics, anti-tumor antibiotics, and anti-amoebic antibiotics. And their chemical structure, for example, beta-lactam antibiotics, tetracyclines, aminoglycosides, macrolides, and so on. We'll be considering classification according to chemical structure as follows. Beta-lactam antibiotics, example, penicillin G, cephalosporin, monobactams, and carbapenems. Aminoglycosides, example, streptomycin, neomycin. Macrolides, example, erythromycin, clarithromycin, and azithromycin. Tetracyclines, example, tetracycline, doxycycline. Lincosamides, example, lincomycin and clindamycin. Polypeptides, example, polymyxins. And miscellaneous antibiotics, example, chloramphenicol. After studying the classification, now we move on to module number 3, which is mechanism of action of antibiotics. The different antibiotics or antibacterial drugs have various targets on the bacteria, including cell wall and cell membranes, ribosomes, nucleic acids, bacterial cellular metabolism, and bacterial cellular enzymes. There are many different mechanisms by which these agents inhibit the multiplication and growth and destruction of bacteria. Antimicrobial drugs exhibit antibacterial effect using one or all of the following mechanisms. First, inhibition of cell membrane synthesis in microorganisms. Example is beta-lactam antibiotics, vancomycin, cycloserine. Second is inhibition of protein synthesis in microorganisms. Example, aminoglycosides, erythromycin, clindamycin, chloramphenicol, and tetracyclines. Third, inhibition or alteration of function of external or cytoplasmic membranes of microorganisms. Example is polymyxin. Now let's move on to module number 4, which is beta-lactam antibiotics. This group of antibiotics is the best known and most widely used and may be divided into two main groups, the penicillins and the cephalosporins. The groups differ only in the nature of the rings appended to the beta-lactam moiety. These drugs were at the forefront of the healthcare revolution of the 20th century, which saw previously incurable and debilitating diseases remedied and controlled. Now let's study penicillins. Penicillin molecules all contain highly strained four-membered beta-lactam ring. fused to a 5-membered thiazolidine ring. The beta-lactam ring is unstable and primarily responsible for the antibiotic potency of these molecules. Synthesis of penicillin analogues. Penicillin analogues were originally synthesized by a fermentation process in which different carboxylic acid derivatives were added to yield penicillins containing different 6-amido derivatives. The principal disadvantage of this approach was that not all carboxylic acids were biologically acceptable, and thus only a limited number of analogues could be prepared. The semi-synthetic approach relies on the isolation of 6-amino penicillinic acid, that is 6-APA, from fermentation media. A large number of 6-amido derivatives can then be prepared by chemical reaction with acyl chlorides. Structural features essential for activity in penicillins. The bicyclic ring system containing the beta-lactam is crucial, as are the cis relationship between the two hydrogens at positions 5 and 6, a free 3-carboxylate and a 6-amide. Changing one or more of these results in a loss of activity. Now let us study the mechanism of action. Penicillins have a structure resemblance to 2 D-alanine residues linked together and are mistaken by the transpeptidase enzyme for D-alanine and thus incorporated into the active site. Once bound, the beta-lactam carbonyl is attacked by the serine hydroxyl and ring opening occurs to leave the penicillin covalently bound to the enzyme. The bulky thiazolidine ring now blocks access to the active site by either a pentaglycine chain or water. As a result, the penicillin becomes irreversibly bound to the transpeptidase enzyme, preventing it from functioning properly. This results in incomplete cell walls that are much more fragile and porous and eventually lead to swelling followed by cell lysis and death. Now let us study what is penicillin resistance. Penicillin resistance first became a serious problem in the early 1960s. Overuse of penicillin, that is particularly penicillin G, led to rapid emergence of penicillin-resistant bacterial strains which contained an enzyme, beta-lactamase, which deactivated the drug by cleaving the beta-lactam ring. Now let us study the resistance mechanism. In the figure, penicillin-resistant bacteria possesses beta-lactamase enzymes which deactivate the drug by opening the beta-lactam ring. Penicillin derivatives like methicillin and neficillin containing bulky groups on the side chains were poor substrates for beta-lactamases and hence less prone to it. Now let us study about cephalosporins. Cephalosporins were the second major group of beta-lactam antibiotics to be discovered. Cephalosporins contain a beta-lactam ring like the penicillins, the main difference being that while the latter contains a 5-membered thiazolidine ring, cephalosporins have a 6-membered dihydrothiazine. Their mode of action against bacteria is essentially the same as that of penicillins. Structural features essential for activity. The development of the cephalosporins is conveniently divided into generations. First-generation cephalosporins, that is house cephalosporins, example, cefazolin parenteral, cefalexin oral. Second-generation cephalosporins, example, cefoxitin, cefotetin and cefuroxim. And third-generation cephalosporins, example, cefotaxime, ceftriaxone and ceftazidime. And fourth-generation example, cefepime. Now let's study about carbapenems. Thinamycin, discovered by Merck in the late 1970s, is one of the broadest spectrum antibiotics ever discovered. It uses import porins unavailable to the other beta-lactams to enter gram-negative bacteria. Due to its high unstable nature, this drug and its derivatives are created through synthesis, not bacterial fermentation. Thinamycin was slightly modified and marked as imipenem. Other modifications of thinamycin have produced superior carbapenems called meropenem and ertapenem, which are not as easily degraded by renal peptidase and do not have the side effects of the imipenem. Now let's study monobactams. The only clinically useful monobactam is aztreonam. While it resembles the other beta-lactam antibiotics and targets the penicillin-binding proteins of bacteria, its mechanism of action is significantly different. It is highly effective in treating gram-negative bacteria and is resistant to many beta-lactamases. Beta-lactam inhibitor combinations: aminopenicillins, ampicillin sulbactam, that is unesin, amoxicillin clavulinate, augmentin, extended-spectrum penicillins, piperacillin, tazobactam, zosyn, ticarcillin, clavulanate, timentin. Now, let's move on to module number 5, which is aminoglycosides. The discovery of streptomycin, the first aminoglycoside antibiotic to be used in chemotherapy, was the result of a planned and deliberate search begun in 1939 and brought to fruition in 1944 by Schatz and Associates. This success stimulated worldwide researches for antibiotics from the actinomycetes and particularly from the genus Streptomyces. Among the many antibiotics isolated from that genus, several are compounds closely related in structure to streptomycin. 6 of them are kanamycin, neomycin, paramomycin, gentamicin, tobramycin, and netlimycin. Currently are marketed in the United States. Amikacin, a semi-synthetic derivative of kanamycin A, has been added, and it is possible that additional aminoglycosides will be introduced in the future. All aminoglycoside antibiotics are absorbed very poorly, less than 1% under normal circumstances, following oral administration, and some of them, that is kanamycin, neomycin, paramomycin, are administered by that route for the treatment of gastrointestinal infections. Because of their potent broad-spectrum antimicrobial activity, they are also used for the treatment of systemic infections. Their undesirable side effects are, particularly ototoxicity and nephrotoxicity, have restricted their systemic use to serious infections or infections caused by bacterial strains resistant to other agents. When administered for systemic infections, aminoglycosides must be given parenterally, usually by intramuscular injection. An additional antibiotic obtained from Streptomyces, spectinomycin, is also an aminoglycoside but differs chemically and microbiologically from other members of the group. Now let's study the mechanism of action. Aminoglycosides act directly on the bacterial ribosome to inhibit the initiation of the protein synthesis and to interfere with the fidelity of translation of the genetic message. They bind to the 30S ribosomal subunit to form a complex that cannot initiate proper amino acid polymerization. They also cause misreading mutations of the genetic code. Chloramphenicol blocks this transport system. The effect of the aminoglycosides is bactericidal and is enhanced by agents that interfere with cell wall synthesis. Resistance. Resistance to aminoglycosides is becoming a problem. It occurs through several different mechanisms, the most important being inactivations by microbial enzymes, of which 9 or more are unknown. Amikacin was purposefully designed as a poor substrate for these enzymes, but some organisms have developed enzymes that inactivate this agent as well. Resistance as a result of failure of penetration can be largely overcome by the concomitant use of penicillin or vancomycin. Antibacterial spectrum. The aminoglycosides are effective against many aerobic, gram-negative and some gram-positive organisms. They are most widely used against gram-negative enteric organisms and in sepsis. They may be given together with penicillin in streptococcal infections caused by Listeria species and Pseudomonas aeruginosa. Gentamicin is the aminoglycoside most commonly used, although tobramycin is a preferred member of this group for Pseudomonas aeruginosa infections. Amikacin has the widest antimicrobial spectrum and along with netlimycin can be effective in infections with organisms resistant to gentamicin and tobramycin. Now let us study the structural activity relationships of aminoglycosides. Aminoglycosides are compounds that contain 2 or more amino sugars joined by glycoside bonds with an aminocyclitol ring, that is aglycone part. The 6-membered aminocyclitol ring is either streptidine, 1,3-diguanidino, 2,4,5,6 tetrahydrocyclohexane, as in streptomycin, or 2-deoxystreptamine, that is 1,3-diamino 4,5,6-trihydroxy cyclohexane, as in all other aminoglycosides. It is convenient to discuss sequentially aminoglycosides SARs in terms of substituents in ring 1, 2 and 3. Ring 1 is crucially important for characteristic broad-spectrum antibacterial activity, and it is the primary target for bacterial inactivating enzymes. Amino functions at 6' and 2' are particularly important, as kanamycin B, which is 6' amino, 2' amino, is more active than kanamycin A, which is 6' amino, 2' hydroxyl, which in turn is more active than kanamycin C, which is 6' hydroxyl, 2' amino. Methylation at either the 6' carbon or the 6' amino positions does not lower appreciably antibacterial activity and confers resistance to enzymatic acetylation, the 6' amino group. Removal of the 3' hydroxyl or the 4' hydroxyl group or both in the kanamycins, example 3' 4' dideoxykanamycin B or diabecasin, does not reduce antibacterial potency. The gentamicins also lack oxygen functions at these positions, as do cecomycin and netelimycin, which also have a 4' 5' double bond. None of these derivatives is inactivated by phosphotransferase enzymes that phosphorylate the 3' hydroxyl groups. Evidently, the 3' phosphorylated derivatives have very low affinity for aminoglycoside binding sites in bacterial ribosomes. Few modifications of ring 2, that is deoxystreptamine, functional groups are possible without appreciable loss of activity in most of the aminoglycosides. The 1 amino group of kanamycin A can be acylated, example amikacin, however, with activity largely retained. Netelimycin, that is 1N-ethyl cecomycin, retains the antibacterial potency of cecomycin and is resistant to several additional bacteria inactivating enzymes. 2 hydroxycecomycins claimed to be resistant to bacterial strains that adenylate the 2' hydroxyl group of ring 3, whereas exhibits good activity against bacterial strains that elaborate 3-acetylating enzymes. Ring 3 functional groups appear to be somewhat less sensitive to structural changes than those of either ring 1 or 2. Although the 2 deoxygentamicins are significantly less active than their 2 hydroxyl counterparts, the 2 amino derivatives, that is seldomycins, are highly active. The 3 amino groups of gentamicins may be primary or secondary with high antibacterial potency. Furthermore, the 4 hydroxyl group may be axial or equatorial with little change in potency. Now let's study about streptomycin. Streptomycin trans 2,4-diguanidino 3,5,6 trihydroxy 5-deoxy 2-ortho 2-deoxy 2-methylamino alpha L glucopyranosyl 3-carbon hydroxyl methyl beta-L-lexo pentofuranoside is isolated from culture liquid of the vital activity of the actinomycetes Streptomyces griseus. Streptomycin has a broad spectrum of antibacterial activity. It is effective with respect to most gram-negative and a few gram-positive bacteria, Staphylococci, Streptococci, Pneumococci, Gonococci, Meningococci, stimulus of dysentery, brucellosis, tuberculosis, rabbit fever, plague, and others. It is used for various diseases caused by microorganisms that are sensitive to bacterial endocarditis, peritonitis, meningitis, infections of the urinary tract, gastritic infections, and so on. Synonyms of this drug are streptan, streptocol, and others. Neomycin. Neomycin is a complex mixture of antibiotics, that is neomycins A, B, C, D, E, and F, that is formed by the actinomycetes Streptomyces fredii. Neomycin A, also called neamine, is 2-deoxy 4-ortho 2,6-diamino 2,6-dideoxy alpha-D glucopyranosyl D-streptamine, and it does not display antibiotic properties. At the same time, neomycin B, ortho 2,6-diamino 2,6-dideoxy alpha-D glucopyranosyl 1,4-ortho 2,6-diamino 2,6-dideoxy beta-L idopyranosyl 1,3-beta-D ribofuranosyl 1,5-2-deoxy D-streptamine differs from neomycin A in the presence of second glycoside residue and exhibits powerful antibacterial activity. Neomycin C differs from neomycin B in the orientation of the amino-methyl group in the neosamine part of the molecule. Neomycin, like streptomycin, has a broad spectrum of antibacterial activity. It is effective with respect to the majority of gram-negative and a few gram-positive bacteria, that is staphylococci, pneumococci, gonococci, meningococci, and stimulants of dysentery. Microorganisms sensitive to neomycin become resistant to a lesser degree than streptomycin. It is used for various gastrointestinal diseases caused by microorganisms sensitive to it, including enteritis, which is caused by microbes that are resistant to antibiotics. However, because of its high oto and nephrotoxicity, its local use is preferred for infected skin diseases, infected wounds, conjunctivitis, keratitis, and others. Kanamycin. Kanamycin, that is ortho-3-amino-3-deoxy-alpha D-glucopyranosyl 1,6-ortho-6-deoxy 6-amino-alpha D-glucopyranosyl 1,4-2-deoxy D-streptamine is isolated from a culture liquid of the actinomycete Streptomyces kanamyceticus, which produces 3 antibiotics, kanamycins A, B, and C. Kanamycin A is similar to streptomycin and neomycins and has a broad spectrum of antimicrobial action. It is active with respect to most gram-positive as well as gram-negative microorganisms, that is staphylococci, gastric bacilli, rabbit fever, Friedlander's bacillus, Proteus, Shigella, Salmonella. It is used for treating sepsis, meningitis, osteomyelitis, peritonitis, pneumonia, pyelonephritis, pyelocystitis, infected wounds, and post-operational purulent complications caused by microorganisms sensitive to the drug. Tobramycin. Tobramycin is ortho-3-amino-3-deoxy-alpha D-glucopyranosyl 1,6-ortho-2,6-amino-2,3,6-trideoxy-alpha D-ribo-glucopyranosyl 1,4-2-deoxy D-streptamine is isolated from a culture liquid of the vital activity of the actinomycete Streptomyces tenebrius. Tobramycin is highly active with respect to gram-negative microorganisms as well as gram-positive microorganisms. It is used for several bacterial infections, peritonitis, sepsis, meningitis, osteomyelitis, endocarditis, pneumonia, pleural emphysema, pulmonary abscess, purulent skin infections, and soft tissue infections, and infections of the urinary tract caused by microorganisms that are sensitive to the drug. Gentamicin. Gentamicin is a complex of antibiotics isolated from a culture liquid of the actinomycetes Empurpurea, which consists of a mixture of approximately equal amounts of 3 compounds, gentamicins C1, C1a, and C2. Gentamicin has a broad spectrum of biological action and is highly active with respect to strains of staphylococci that are resistant to penicillins and other antibiotics, many gram-negative microorganisms. It is used for pyelonephritis, cystitis, pneumonia, pleural empyema, peritonitis, sepsis, meningitis, purulent skin and soft tissue infections, infected wounds, burns, and so on, which are caused by microorganisms that are sensitive to the drug. Module 6. Tetracyclines. Tetracyclines were discovered in the 1940s and exhibited activity against a wide range of microorganisms including gram-positive and gram-negative bacteria, chlamydia, mycoplasmas, rickettsia, and protozoan parasites. Tetracyclines are a class of chemically similar drugs that are unified by the presence of 4 rings joined in hydronaphthalene system, and the differences in activity are determined by various substituents on the base structure. Mechanism of action. Tetracyclines exert their bacteriostatic effect by inhibiting protein synthesis in bacteria. This antibiotic prevents transfer RNA molecules, a type of nucleic acids which transport amino acids, from binding to the 30S subunit of bacterial ribosomes. It is possible for tetracyclines to inhibit protein synthesis in the eukaryotic cells of the host, but the drug is less likely to reach the required concentrations in humans because eukaryotic cells do not have a tetracycline uptake mechanism. Antimicrobial spectrum of tetracyclines. Tetracyclines are broad-spectrum antibiotics which exhibit activity against a wide range of microbes, including both gram-positive and gram-negative bacteria, chlamydias, mycoplasmas, rickettsia, as well as some protozoan parasites. Now let us study the SAR of tetracyclines. Tetracycline molecules comprise a linear fused tetracyclic nucleus, rings designated as A, B, C, and D, to which a variety of functional groups are attached. The simplest tetracycline to display detectable antibacterial activity is 6-deoxy 6-D-methyl tetracycline, and so this structure may be regarded as a minimum pharmacophore. Features important for antibacterial activity among the tetracyclines are maintenance of the linear fused tetracycle, naturally occurring stereochemical configurations at the 4A, 12A, that is AB ring junction, and 4-dimethyl amino group positions, and conservations of the keto-enol system at positions 11, 12, and 12A in proximity to the phenolic G ring. Now let's study about chlortetracycline. It is 7-chloro-4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A oxtetrahydro 3, 6, 10, 12, 12A pentahydroxy 6-methyl 1,11 dioxo 2-naphthalene carboxamide is obtained biosynthetically as a result of the activity of a microorganism, the actinomycetes Streptomyces aureofaciens. Oxytetracyclines. Oxytetracycline, that is 4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 3, 6, 10, 12, 12A hexahydroxy 6-methyl 1,11 dioxo 2-naphthalene carboxamide is synthesized biosynthetically as a result of the activity of actinomycetes Streptomyces rhinosus. Tetracycline. Tetracycline, that is 4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 3, 6, 10, 12, 12A pentahydroxy 6-methyl 1,11 dioxo 2-naphthalene carboxamide is synthesized by reducing chlortetracycline with hydrogen using a palladium on carbon catalyst. However, it can be synthesized microbiologically using the actinomycetes Streptomyces viridifaciens as well as certain mutant Streptomyces aureofaciens. Demiclocycline. It is 7-chloro-4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 3, 6, 10, 12, 12A pentahydroxy 1,11 dioxo 2-naphthalene carboxamide is produced by a mutant strain of Streptomyces aureofaciens. Methacycline. Methacycline is 4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 3, 6, 10, 12, 12A pentahydroxy 6-methylene 1,11 dioxo 2-naphthalene carboxamide is synthesized from oxytetracycline, which is reacted with a sulfur trioxine pyridine complex, resulting in an oxidation reaction. Doxycycline. Doxycycline, that is 4-dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 3, 5, 10, 12, 12A pentahydroxy 6-methyl 1,11 dioxo 2-naphthalene carboxamide is an isomer of tetracycline that differs only in the placement of one hydroxyl group. Minocycline. Minocycline is 4,7-bis dimethylamino 1,4,4A, 5, 5A, 6, 11, 12A octahydro 310, 12, 12A, tetrahydroxy, 111, dioxo, 2-naphthalene carboxamide is synthesized from 6-dimethyl tetracycline, which is synthesized as a result of the vital activity of Streptomyces aureofaciens. Now let's proceed to the module number 7, which is miscellaneous antibiotics. Chloramphenicol, which is D-3O-22-dichloro, N-beta-hydroxy-alpha, hydroxymethyl N-nitrophenylacetamide was first isolated in 1947 from culture fluid of the actinomycetes Streptomyces venezuelae. However, it is only currently produced synthetically. Now let's study the mechanism of action. Chloramphenicol is a bacteriostatic by inhibiting protein synthesis. It prevents protein chain elongation by inhibiting the peptidyl transferase activity of the bacterial ribosome. It specifically binds A2452 residues in the 23S ribosomal RNA of the 50S ribosomal subunit, preventing peptide bond formation. While chloramphenicol and the macrolide class of antibiotics both interact with ribosomes, chloramphenicol is not a macrolide. It directly interferes with substrate binding, whereas macrolides sterically block the progression of the growing peptide. Spectrum. Chloramphenicol has a broad spectrum of antimicrobial activity including gram-positive, gram-negative, aerobic and anaerobic bacteria, Spirocheta, Mycoplasma, Chlamydia and so on. However, it can cause pronounced suppression of blood flow, which is accompanied by reticulocytopenia, granulocytopenia and, in severe cases, aplastic anemia. With this, we come to the end of our episode. Now let's study the summary. The word antibiotic literally means against life. The goal is to kill or stop growth of these tiny infection-causing organisms known as pathogens. There are different types of antibiotics either produced from microorganisms directly or being semi-synthetically. Each category has its own way of killing or inhibiting organism. Figure in above text clearly defines the different mechanisms of actions of various category of antibiotics. There are antibiotics like penicillin, which inhibits cell wall synthesis of pathogens, while antibiotics like macrolides and aminoglycosides act by stopping protein synthesis, hence killing or inhibiting pathogens. Similarly, based on their mechanism of action, macrolides, tetracyclines, and chloramphenicol are widely known as bacteriostatic in nature, while aminoglycosides and beta-lactams are well-known bactericidal agents. A special structure identified in each category is masked for its activity. For example, beta-lactam ring in penicillin and amino sugar substructure in aminoglycosides. Penicillin and macrolides are top 2 prescribed antibiotics. Pathogens are also posing challenging by evolving different resistant mechanism to inhibit mechanism of antibiotic activities. Chloramphenicol and tetracycline, though cheaper and widely used antibiotic, are no longer prescribed due to increase in bacterial resistance. Research work is evolving with inventions and discovery of modern antibiotics which can serve dual purpose of being broad spectrum as well as counter the bacterial resistance mechanism.